So today's webinar is presented by Uwe Rascher, and I'm gonna hand over the word to him now. So we will switch to his computer. So Uwe, if you are ready, here it comes.
So thanks. I think I'm now the presenter, and thanks for the introduction, Rick, and thanks for inviting me and giving me the chance to present here in this webinar. So I still have to switch a few of the slides as we tested this morning, but I think now you should see my first.
Yep, now it's okay. I can see not the presenter view, but the slides you want to present and share with us. Yep.
I will now turn down a little bit my volume because of the background announcements. If there's anything, as Rick said, if there are any questions, if there are things which are unclear, which we want to discuss, post it in the chat window, and at the end, Rick will combine this, and I think we have time for the discussion at the end. And for today's webinar, I would like to take you actually on a little journey from small-scale processes which are related to photosynthesis and end at the end on the satellite level. And I hope by going over these spatial scales from the laboratory to the satellite, I can convince you, I can show you how we can actually quantify photosynthesis by means of fluorescence measurements, and how we can use this not only for phenotyping, but more generally also for ecosystem monitoring and other approaches. So I now try also my laser pointer. I hope this works. And as I already introduced us, I would like to talk with you about photosynthesis. And I think here in this group, we have a pretty mixed group of people coming from plant sciences, people coming more from image processing. So I think everybody has a rough understanding of photosynthesis. So photosynthesis is the process all green plants perform. They absorb the light of the sun, use the energy of the sunlight to fix carbon dioxide, and at this whole process, they release oxygen by the splitting of water. And this process of photosynthesis, I think everybody knows from the textbook, is one of the central metabolic pathways which fools all the processes on Earth, where plants gather the energy to produce biomass, which is then used for our products which we make from plants. But also from the other side, as it's a really central process, photosynthesis is a key process which is also affected by pests, diseases, by extreme environmental conditions, or also by our management of our crops. And therefore, photosynthesis is one of those metabolic pathways which is in the focus of research for decades already. And we know that photosynthesis is not a dynamic process. It changes depending on the environmental conditions. And we also know that photosynthesis, being the central pathway, is also being affected by the effects which we have and which we expect on in times of global change. And therefore, what you see there, it's one of the main research focus that we want to develop a better understanding of the dynamic nature of photosynthesis to, at the end, if you want to come from the user side, to protect and sustain our Earth vegetation and productability. And when I said at the beginning, I would like to take you on a journey from the small-scale processes, I would like to dive in with you in this organs first, where photosynthesis actually happens. Photosynthesis takes place inside the leaves. And for the non-plant biologists, inside the leaves, you have the plant cells. And in the single cells, we have additional organs, the so-called chloroplasts, which have two membranes. And the inner membrane is stacked, looks like coin stacks. And within these membranes, we have the pigments, the green pigments, mainly chlorophyll and others, which are the main pigments which absorb light of the sun and where the process of photosynthesis takes place. But even though if we zoom a little bit further into these pigment clusters, those pigments, which you see here as a 3D representation of the pigment structure of one photosystem, this pigment structure is pretty complex. Different molecules hold the pigments in the exact place, and in a normal photosynthetic machinery, approximately 400 chlorophyll pigments are held exactly in the right position to harvest light in a so-called antenna complex and to shift the energy to the core complexes, which you see here, D1 and D2. These are the core complexes where then the plants are able to use this energy in principle. You don't have to know this in detail, and we're also not going into a plant physiological detailed lecture, but you should know that this process happens on a really, really small scale. And when we speak about photosynthesis, which happens in leaf, going back to the scheme which I had before, that inside these leaves, there's a lot of regulation going on how these pigments are actually exposed to light and how this energy of the incoming photons is harvested to finally be used as the main energy source for plant metabolism. And in this first process, one of the first processes is shown here in a little bit different schematic representation. So we have the pigments, light comes in by the photons, and this energy which is used by the plants absorbs the photons and shuffles some electrons in a pretty complex metabolic pathway. And why I point out this, during this absorption of light in this pigments of the photosynthetic apparatus, there is a weak red light, which is also emitted from these core elements where light is absorbed for the first time. And this red light is the so-called fluorescence light, which you now can see also in the other slide. So what you see here is a solution of green pigments of intact photosynthetic machinery. And if you shine blue light on the solution, and if you carefully look from the side, you see a red light, which is emitted as a red glow from this photosynthetic apparatus. And if you look on this red glow, if we think in terms of a little bit more physical part, if this is the wavelength, this is the red spectrum of our light, then we see that this emitted light has actually two features, one in the red and the other feature in the far red, which corresponds also to the two main clusters of the photosystems, photosystem 2 and photosystem 1, which emit this fluorescence light and produce this red weak glow of every photosynthetically active organism. So continuing in this direction, this red light is pretty weak. Normally you cannot see it, but it's really existing. And we have made in the past years also quite some effort to actually develop some easy approaches where you use colored goggles, some filter glasses, some specifically blue light to actually make this red glow of photosynthesis or this red glow of plants also visible to the general public, to pupils, to students, that convince a wider audience that this red light is really always emitted and that plants are not only green, but the plants also glow red. So why I'm telling you this? This red light has another specific feature. Namely, if you go back to the scheme which I showed you before. So light is absorbed by the photosystems, then shuffled to the reaction centers, and from these reaction centers, normally the energy is used to drive photosynthesis. However, this process of light capturing and light shuffling and usage is never complete. It has a maximum efficiency of up to 83%, but some part of this energy which cannot be used in the process of photosynthesis, some part of this energy is re-emitted as the fluorescence light. And as this is a re-emitted light, the fluorescence light changes depending on the status of the energization of the reaction centers in the photosynthetic machinery. So you can picture if the plant is exposed to high light conditions, if more energy arrives in the reaction centers, if there's more energy available than actually can be used in the process of photosynthesis, then this fluorescence signal increases. And having said this, by looking in this fluorescent signal and by measuring this red glow, we can have a direct measure and a direct grip on what actually happens in the core complexes of photosynthesis, and we can estimate of how much energy is available which cannot be used in photosynthesis. And now I would like to skip about 20 or 30 years of research, because this was already discovered in the late 80s. And in the meantime, plant biologists like this light a lot. So by little clip-on devices, and this is just one example which I show you here on this slide, we can measure this red fluorescence light for science, and by applying sometimes a little bit complex measurement protocols, we have a means to actually measure non-invasively the efficiency and the status of the photosynthetic machinery by just measuring this red glow. In a few years, I have made a literature research, and at the moment there are approximately 750 papers published in really the scientific literature which use this method to get an idea of the status of photosynthesis. And these papers, of course, go from a lot of laboratory work, greenhouse work, basic plant research work. And one disadvantage of the main measurement protocol for years was that to assess this red light, you normally had to be pretty close to the leaf. You had to apply specific light pulses, you had to manipulate the photosynthetic machinery to then get reliable parameters out of measurement protocols, which are exemplified here in this slide. And already about almost 20 years ago, 15 years ago, we started some first approaches to develop some instruments which also are able to measure this fluorescence light, this red glow, also from some distance. And this is a slide or a picture from 2014, where one of those first monsters, which is a big laser and a big telescope, which we mounted on a tower, and with which we could shoot a laser light on experimental plots in our agricultural site, and where we could detect this fluorescence light from quite some distance. This instrument was still pretty big and a little bulky, and in the years we realized that it makes more sense to have a lighter instrument, which you now can see here, on the compromise that we may have to be a little bit closer to the canopy. And in a consortium with several other people, and also some colleagues from Australia were involved, and Barry Osmond was one of the drivers behind this. We have developed an instrument which is called LIFT, Light-Induced Fluorescence Transient, which can now be mounted on plant phenotyping platforms, and with which we can drive over the field, and where we can actually measure on the run by just driving over the field the fluorescence light, and where we can relate this to the actual status of photosynthesis. There are a few pictures from our field trial, and a PhD student who was measuring at Maricopa at a big breeders trial. In the back you see the big LenderTech system just being set up at this time, and we were running with our light lift laser over this field, and there we learned that we can measure up to 1,000 plots in one day by just running over the canopy and being able to measure the transients and the dynamic transients of this fluorescent signal to relate this to the status of photosynthesis. So fluorescence measurements, kind of a established and successful method on the clip-on with clip-on devices, also now almost operational and being sold for top of canopy measurements. But with these instruments, we still are facing the problem that we have an active sensor where we have to apply a measurement light, an active measurement light, and where we detect the emission of this fluorescence light then from, in our case, up to 2 to 3 meters distance. And when I was saying at the beginning, I want to take you up to a satellite mission for larger scale measurements and for even passive measurements, it would be desirable to also be able to measure passively, so to not have the necessity to shoot an active, pretty strong measurement light on the plant canopy. And this would be the second part of my talk, with which I would like to take you now. And again, I would briefly like to take you back to the thoughts of a Russian engineer in the 17th, who already realized that if we look on the one hand on the emission of the fluorescence light, which is the green curve in this case. And if we also consider the transmissivity of our Earth atmosphere, which is the blue curve in this case, that the emission of the fluorescence light just by chance falls in line with two big absorption lines in our Earth atmosphere. And these two absorption lines are a result of the oxygen in our Earth atmosphere. And the atmospheric oxygen in our atmosphere absorbs photons here in the red and in the far red region. And most of the photons which come from the sun by penetrating through the Earth atmosphere are actually absorbed. And if we measure with high spectral resolution on the ground, in this small spectral windows, we have a greatly reduced or almost zero incoming flux of photons of this specific wavelength. These features are pretty small, but nevertheless, if you can resolve those features, you should be, and this was the theory of the 17th, be able to see this fluorescent signal, because the fluorescent signal is not a reflected light, but it's a glow. It's an inherent glow coming from the photosynthetic machinery. So this glow happens independent of the incoming light. If we shine blue light on a leaf, red fluorescence is emitted. So, and this means that the relative or the apparent reflectance is actually a product of the reflected light plus the emitted light, and the emitted light or the measurement signal is greatly enhanced, or the contribution of the fluorescence light is greatly enhanced in those atmospheric absorption lines. For those who have not fully followed me so far, a little bit differently. So you can also see this a little bit more schematically. You have incoming light at one spectral regions. Some photons are absorbed by the atmosphere. And then you have an additive signal, which is this broadband fluorescent signal, which is emitted and which shifts both the reflectance or the measured light additively up. And this additive shift is, of course, different, or which can be resolved if you compare the differences in the incoming light across those absorption lights. And a little bit schematically, you can also say this: if you compare the energy or the light which you measure outside and inside of those atmospheric absorption bands, you can, with some mathematical retrieval, you can separate the reflected from the fluorescence contribution to your measurement signal. And this was a method, so-called Fraunhofer line detection, already discovered in the 70s, described in Russian, and then forgotten again for 15 years, and then revisited at the beginning of the 19th by some American and European scientists. And in this meantime, the technology for high-resolution spectrometers, which allow us to really resolve these nanometer or sub-nanometer resolution light spectra, we were now able in the past years to actually really develop the measurement instruments which consist of high-resolution spectrometers, which are built in a reasonably good stabilized box, and by measuring the incoming light and the outcoming light over canopies, by using then this retrieval methods of the Fraunhofer line retrieval, we can now use this high resolution spectrometers to actually measure this emitted fluorescence light in these two spectral regions, the red and the far red region, on top of canopies. And this was a research which was started pretty much 5 years ago. We, together with the University of Milano, have also developed the first product. There's now a second product on the market from the American partners. And at the moment, we are facing the situation that approximately 35 to 40 continuous measurement stations are employed worldwide, where people measure on top of the canopy. This is Spain. This is an area at our side where people measure continuously the emitted fluorescent signal to get an understanding of the temporal, seasonal dynamics and adaptation of the photosynthesis of the vegetation below. And this was, as I said, that's an ongoing global initiative where people buy these boxes, employ those on their ecosystems. But I would also like to draw you, by using these point sensors, But then we also had another idea and said, well, if it's only red light which we measure, why not using an imaging spectrometer, a high-resolution imaging spectrometer, to also use the same measurement principle and to measure solar-induced fluorescence under natural conditions in an imaging mode. And this is a picture from also about 3 to 4 years ago, one of our spectrometer, one of my PhD students sitting on top of a sugar beet canopy. And by doing this measurement precise enough and by spending quite some time in the retrieval, means in the separation of the fluorescence light from the background reflectance, we finally could resolve or also map the intensity of solar-induced fluorescence under fully natural conditions in a natural canopy. And we could do this just by passively using the sun as the exciting light. By doing so, one of the first publications was that when we then follow, for example, the yield of solar-induced fluorescence, so this is already corrected for the changes in light intensity during the day, and we only look on those leaves which are the outer leaves and which are fully exposed to the sun, then we realize that with the increasing light intensity, the fluorescence increases in the morning. This is expected. More light, more saturation of the photosynthetic machinery, more electrons which cannot be used photosynthetically, and the fluorescence light increases. But then during midday, we realize the light intensity further increases, but the fluorescence signal decreases. And only later in the afternoon, starting this time at 2 to 3 o'clock, then we see a recovery of the fluorescent signal with a further decreasing light intensities. So this was one of the surprising things, which we then, however, with the work of other colleagues reproduced by other groups, we could also make sense out of it by realizing that this photosynthetic yield and the fluorescence yield is not just a simple one-to-one linear correlation, but that by using our knowledge and expanding of what I presented you before, that plants are not stupid. And going back to my scheme, which I showed you before, if I have an over-energization of the photosynthetic machinery, and if I have a block of photosynthesis, or not all the energy can be used in photosynthesis, of course, in the first instance, the fluorescent signal increases. This is the model which I explained to you before. But after some while, after some sustained over-energization of the photosynthetic machinery, the plant normally activates a big variety of different protection mechanisms, which are also able to dissipate the additional energy which cannot be used photosynthetically. These energy or these processes are normally summarized as heat or non-photochemical quenching processes, and these processes then also dissipate the energy, and therefore the fluorescent signal decreases again. And this is exactly what we see and what we also know now is being established in the community, that we have a different domains, where we have a domain where only the photosynthetic apparatus is dominating. This is a little bit hard to read. So this is low light. With increasing light, the fluorescent signal increases, but this is still no additional regulation is necessary. Then we have one path where actually this additional non-photochemical quenching mechanisms kick in and the fluorescence light decreases again. And if we finally go to severe stress conditions or photodestruction, where all the dynamic protection mechanisms of the plants do not work again, then the fluorescent signal increases and partly increases pretty dramatically. So this is then really the domain of severe destruction of photoinhibition. And with this back in the back of your head, I would like now to take you from the top of canopy scale where I just arrived to the aircraft level. And the measurement principle, which I showed you before, which you could do on top of canopy, in principle also can be applied on the larger scale. And years ago, we started to develop the first airborne sensor, which at this time was the only one worldwide, where we had a big spectrometer, a highly controlled spectrometer, and a big research aircraft. And with this spectrometer, we could resolve the spectral reflectance of our surface in the visible range, but also expand it to the near and shortwave infrared. This is this module. And we had a custom-built second module where exactly in the spectral region of the red and the far-edge spectrum, the emitted reflectance of the surface was resolved in high spectral resolution. And what I showed you before with these two oxygen absorption bands, these two absorption bands are also being seen here. And these are real airborne data. So these are measurements from a flying aircraft flying over real vegetation. And you can see that these absorption lines, these atmospheric absorption lines, which I presented before, are really well resolved also in this airborne data. For people who are interested in here, these are water absorption bands, which are not really useful for the retrieval because the water vapor in the atmosphere changes a lot. So, but with this airborne sensor, we could actually really map or use the same principle for fluorescence measurements as we had on the ground. However, knowing this is an image processing group, I want to put in a few slides about what this actually means. This processing of the data of a flying aircraft, which is a wobbly and non-stable platform, where we also have a specific atmosphere underneath the aircraft and the surface, which of course also influences the signal, and where we have to correct for all these properties to resolve this weak red glow. We had to develop a pretty complex processing pipeline to actually work on the data, on this airborne data. And this is just a snapshot of a work of a lot of image processing, image retrieval, work on the data, knowing the sensor correctly, including knowing, for example, for people who have fun in this, for example, to knowing the point spread function of such a sensor very precisely, why such a point spread function is a little bit different than the normal point spread function, because it's mirrored on the spectrometer in a spatial and a spectral domain on the same detector. So it's not a normal optics, but it's a specific slit which produces this image. Nevertheless, we know if you want to measure it precisely, you have to correct for this point spread function. And all these parts had to be included in a correct processing of this airborne data. And if you have fun of this, so the people from the imaging community, they are more than invited to read a paper which we just have in print in remote sensing. It should come out every day. I was checking just a minute before because I expected really every day to come out, but it's not there yet. But as I said, there's quite some who have fun with image processing, hardcore image processing behind, until we reach really the point where we then can work again with the data, means at sensor radiance, relative reflectances, and then also the traits in terms of vegetation indices or other parameters which we would like to have if we fly over those landscapes. And in addition to this airborne sensor, we now also add the fluorescence, the solar-induced fluorescence, which we can measure at the moment because it's still in the research project with different retrieval methods and being able to separate the red and the far-red fluorescence. So having an idea of this two-peak feature of our signal. And this is something where I now would like to give you just an idea, a snapshot idea of what we have done with our aircraft sensor in the past. This is the first image which we retrieved from our research campus at Campus Klein-Altendorf. So these are big fields, apple plantations, experimental plots. And what you can immediately see is that this fluorescent signal is not just a representation of the greenness, because it represents the amount of chlorophyll in the first instance, but then also the activity of the photosynthetic machinery, which modulates the fluorescent signal. This was then something which in the 2014-15 we tested in the Czech Republic in one of the first campaigns, where we used a simple ecosystem, a lawn which we bought, which we built up reasonably big that we could see it from the aircraft, and we applied a herbicide, DCMU, and this herbicide is known from laboratory studies that it blocks photosynthesis completely, And by blocking this photosynthesis, we know from our laboratory studies that this should increase a strong fluorescent signal without really destroying the pigment. So it's really a physiological block. And this is exactly what we have seen there. So we applied the DCMU on one of the treatment plots, and we flew over this 2 hours later, and we could see that the fluorescent signal really increased depending on the dose which we gave, and this was not associated with a breakdown of the pigments. So this was really a look into the functional status of the photosynthetic machinery. And based on this, our data are currently used in a variety of different collaborative projects. First projects came from people who were interested in getting new methods to monitor carbon dynamics in fields. GPP, gross primary productivity, is the measurement of the carbon uptake by green vegetation. And there the group around Bastian Wienicke could show for the first time that when we include this fluorescent signal, which is seen here in the prediction of the GPP, that we can actually really increase the predictions of the carbon models. And this is something which is now currently also being picked up by various studies, also on the larger scales, using various data to have a direct measurement of photosynthesis by means of this fluorescent signal. In this time, we were having focused a lot on better process understanding to really understand the meaning of this fluorescent signal. And in 2 recent papers, we did some of the experimental treatments, which we also know on the small scale from the laboratory. We applied this in the field. We have set up several field plots where we had a specific treatment which would represent or could represent natural stresses and environmental conditions to see how really this fluorescent signal changes quantitatively. We were working with breeders and different plant varieties which have a specific change in the photosynthetic machinery. This, for example, is a soybean mutant which has a greatly reduced chlorophyll content by still maintaining almost the full yield and full photosynthetic capacity. And this was something where we say, okay, fewer chlorophylls, but still the same amount of photosynthesis should be detectable in the fluorescent signal, which you could also show. And at the end, we are now also going in the natural monitoring. This is our field site with different field plots where we use the combination of the fluorescent signal and vegetation indices and temperature measurements to predict the effect of natural occurring heat waves. Or another study, I don't go in the detail, it's really more for people who are interested to read more, so you have all the citations. This is something where we look on a field where we know that we have different water availability because of different rooting depth and different water holding capacity in the soil, and where we could also see how we can actually detect this different water availability in plants which affects photosynthesis via the fluorescent signal. We are not only working with crop plants, but we are working a lot with man-made plants. One study comes from the forestry community where you could show that the fluorescent signal, especially the red one, is linked to tree age, which has something to do with a different physiology of younger needles to older needles. And as I said before, we are now trying to use this fluorescent signal, which we are confident to retrieve, to implement it in different models, larger scale models, either on the regional scale or on the larger scale, to predict really the carbon uptake of vegetation. And with this, I think I'm almost at the end. I just would like to give you an idea that what we are doing here, especially the larger flying measurements, are out of the scope of normal plant phenotyping, even though we are still working in a phenotyping community, but this is also linked to the larger scale ecosystem monitoring. And in now 3 years ago, the European Space Agency, ESA, selected a new satellite mission, which is called FLEX. And this will be a new satellite mission which will measure solely, or which is geared towards measuring this fluorescent signal on a global scale. And this will be a mission which will be, or is currently being implemented in the whole path of ESA's Earth Explorer missions. So there are meteorological satellites, there are the operational Copernicus satellites, which you may know, these are the Sentinel families. And there's a branch of new science instrument, and FLEX will become the number 8 in this fleet to be launched at the moment with a time stamp of 2024. So if you hear some news in 2024, maybe you can remember this webinar you were listening today. With our FLEX satellite, we will follow one operational satellite, which is Sentinel-3, which is already delivering data. And this is also a novum of this mission, that we combine the data flow on an early stage between a novel research satellite with an operational Earth Explorer satellite. The main goal behind that is that we want to combine our high-resolution measurements in the oxygen bands, so the same measurement principle which I showed you before. We want to combine this with operational measurements across the spectral window and also the thermal window, which is provided by the Sentinel instrument, already operational. And by doing so, we will, or we at the moment, we are pretty confident that we will be able in 4 to 5 years from now to deliver global maps on a 300 by 300 meter pixel resolution and a revisiting time of 27 days at the equator, which results in a revisiting time in Germany. It will be approximately 12 to 13 days. So if you are more interested in this FLEX satellite mission, knowing that's not the main focus of this community here, but feel free to visit the webpage of ESA. So there's a lot of information also available at ESA's webpage and in the papers which I just showed you in the presentation. And with this, Rick told me I should aim for approximately 30 to 40 minutes. I think I'm now at 36 minutes. That's okay. So I hope I could give you an idea of how, in my perspective, field phenotyping and remote sensing can come together. The example of measuring photosynthesis, which is for me a dynamic but really important trait. Active measurements on the proximity are established and being further exploited and now being complemented by this passive solar-induced fluorescence approaches, which gives the big advantage of being scalable, so we can apply this also on an imaging mode on larger scale. This knowledge of photosynthesis, from my perspective, is crucial for the classical phenotyping approaches, for breeding, for plant management, but also for ecosystem monitoring. And I'm sometimes asked what's more important, ground-based or satellite measurements. For me, it's not the one or the other, but the different approaches across the scales, I think, are both necessary to really have the right data set to link the mechanisms, the dynamics and the dynamic which happen on the small scale with a larger scale monitoring. So with this, I would be at the end. I would like to thank my group who helped me with all this work, who have a lot of fun with bringing all the instruments to the field, where we still have the plants in the center. I would like to take the chance, if you know somebody who is interested in this topic, I currently have 2 positions open, one on the postdoc level, one on the PhD level. So feel free to check out the web pages of the Forschungszentrum Jülich. And last but not least, I would like to thank all the partners who contributed to this work, because making a satellite and building a satellite is for sure a collaborative effort in Europe, but also with some American partners. So I want to thank them all, and of course also the funding agencies who provided the money for all these non-cheap toys and campaigns. And with this, I thank you all for your attention, and I think now we still have time for some questions. Thank you.
Good, thank you. I'm the only one then with a microphone on, so you can hear me, right?
I can hear you, yes.
Good. Okay. Well, we have at least 2 questions, but more questions could still be entered at the chat box. About this lift sensor. So we have a telescope and we have a laser as a light source.
Yes.
If I understand it completely correct.
Yes.
This alignment of the light source and the measurements 20 meters away must be extremely accurate, because you need to watch exactly where the laser is
Yes.
hitting a leaf.
Yes.
Is it a very accurate calibration, or is it how complex is it?
Yes. So thanks. Interesting question. In the first instrument which we had the telescope. Can you still see my slides?
Yep. In presenter mode, actually. Yeah.
Okay. In presenter. Okay. That's fine. I think that's fine. So you can still hang on. I take the pointer again. So we had the telescope for detecting, and back there was the detector, and the laser was somewhere outside of the instrument. And this is exactly the background of the question. We had to align this pretty precisely, and of course this was distance dependent. We could only align those to a specific distance. So in the upgraded version, which we have now here, we are using the same optical path. So we have a big lens in front of the instrument for measuring the fluorescence signal. And through the same lens, we have different fiber optics leading in, and we feed the laser in the same optical path by different beam splitters and light splitters. So we again make use of the different spectral properties. So we have different spectral responses light splitters, and we actually shoot the laser exactly through the same optical path as we measure the fluorescence light back to the instrument. And therefore we are overcoming the problem of the alignment.
Okay, great. Okay. The second question is on the fact that you have the sun-induced fluorescence measurements, but over the day you have changing light conditions.
Yep.
And you have the change of the photosynthesis of a plant anyway over the day.
Yep.
So if you cover large areas, it takes time. During the day, all kinds of changes influence your measurements. So how do you then compare one part of your field with another part of the field?
Yes, totally right. So somebody paid well attention, and I tried to kind of sneak over this point. At this site, I said, well, here we are plotting fluorescence yield. And this fluorescence yield is exactly the way which we try to use to address this problem. Because you're totally right, with a diurnal course, the more light we have, the higher the fluorescence signal comes, which is just a factor of the increasing light intensity. And then, of course, also the fluorescence signal increases. But we would like to know what is really the fluorescence yield, means the fluorescence which is emitted per light or per photon which is absorbed. Therefore, we are normally trying to use the fluorescence yield as a parameter, which is the intensity of the fluorescent signal divided by the total amount of photons which are absorbed by green plant material. And this second part, this estimation of how much light is really absorbed in a canopy, this is a research question by itself. And we sometimes just say we scale it based on the above canopy incoming light. And sometimes we use the whole spectral information to get an estimate of how much light is available. And then we calculate this fluorescence yield parameter, which is then at the end independent of changing environmental conditions or changing absorbance or availability of light energy. And with this fluorescence yield, then we are really close to the efficiency of the photosynthesis. But somebody really paid well attention, because I tried to sneak over this without mentioning this in detail during the talk.
Okay, then a follow-up question. And when you look at this image, then you can see all kinds of differences within one plant already. So you somehow need to aggregate that on plot level. So we take that's already a statistical approach in just taking an average or taking a peak zone value. So this all makes it quite challenging still, is if the ground coverage is different.
Yeah. Mm-hmm. Yes.
How do you deal with those like multidimensional aspects in drawing conclusions?
Yes, exactly. And this is exactly one of the part where I'm at the moment trying to raise the ability. So the first papers which were published after we could measure fluorescence, people just try to develop linear correlations. Higher fluorescence, higher photosynthesis, or maybe down negative correlations. And in the meantime, we know that the simple linear correlations do not really hold true because there are a lot of confounding canopy effects, a lot of confounding effects, how really this signal, which has a big diversity, if you look on a sunlit leaf, on a shaded leaf, how this actually mixes. And we have a lot of nonlinear effects, how this mixing happens. And then at the moment, there are two answers. The one thing is that we have in the meantime, a full modeling approach in hand. So if you look, for example, on this paper, there is a model behind which really allows an integrated view from the leaf level processes. So there's really a fun camera process model linked to a leaf scale radiative transfer model linked to a canopy scale radiative transfer model. This model is the so-called scope model, which was developed by several groups in the past 10 years, I would say, and which fully integrates the fluorescence from the photosystems to the top of canopy level. This model, in principle, is also fully invertible, and there's quite some work from the University of Valencia to use this modeling approach to actually also come down or to link top of canopy measurements to the physiological emission of fluorescent signal at the cell level or at the leaf level. So there's a full modeling approach available. And based on this modeling approach, we are currently developing easier ways, which are empirical or scale down approaches to use the correlations which nevertheless exist. Also, if you move up to the canopy level, if you make some of the corrections right, you can also use this fluorescent signal for extrapolating or determining some functional properties without going in the full modeling approach. Nevertheless, I need to stress the information content of this fluorescent signal, of the solar-induced fluorescent signal, of course, is influenced by quite a lot of those other parameters. And when you want to have a precise measurement of your photosynthetic properties, you have to measure with an active measurement. You have to be close to your plant. So we will never get the full information content of these measurements by measuring from an aircraft. But nevertheless, there's quite a lot of links still existing and also using this modeling approach.
Yeah.
Long answer, but I think.
Yeah, so I can imagine that there are also variety differences. Canopy architecture aspects, all kinds of challenges that need to be overcome. Okay.
Exactly, and in such a model, for example, the different canopy effects, like different leaf properties, different leaf distribution, clumping of leaves has a pretty big influence on the top of canopy signal.
Yeah, exactly. Okay. So if we don't have more questions than that, then I would like to really thank you for giving this webinar. I found it very interesting. And that's my personal opinion. Hopefully the others agree with me. I will, so thank you. I'll do the clapping for the rest of us.
Thank you.
And I will close this session for now. And so everybody that watched live, please also share the word that this has been recorded and will be placed at our IPPN website soon. Okay, thank you everybody, and have a nice day.
Okay, thank you.